Vande Gujarat.
Good afternoon, dear aspirants of the NET. Today, myself, Dr. Amit Ballani, Assistant Professor, Department of Biotechnology from N.V. Patel College of Pure and Applied Sciences, Vallabh Vidyanagar, is going to discuss about cell cycle and its regulation. I have divided my lecture into two parts. In the first part, we'll be discussing about the overall cell cycle, and then in the part 2, we'll be discussing about the various regulatory factors that regulates the cell cycle. As far as cell cycle is concerned, it is very much mandatory to know what are the cells. As we know, the cells are the structural and functional units of life. And all the organisms, the living organisms, the biodiversity we see, all organisms consist of cells, and these cells arise from pre-existing cells. This theory was first of all given by Rudolf Virchow. As far as cell division is concerned, it is estimated that more than 225 billion cells are produced every day by an average human body or average human individual. Each dividing cell is called as mother cell, and its descendants are called as daughter cells. As far as cell division is concerned, it's not just about increasing the number. Cell division links parents to their offsprings and living species to their extinct ancestors. We humans and all multicellular species that we see got here through cell division only. The entire story of an individual actually starts from a single cell that is known as zygote. Once an egg becomes fertilized, cellular division starts, and that gives rise to entire individual. By the process of division and differentiation, we see the development of entire individual. Now, broadly, there are two types of cell division. One is known as mitosis and another is known as meiosis. The meiosis, which is also known as reductional division, is the process by which gametes are generated for sexual reproduction. Here, a diploid cell after meiotic division would give rise to 4 haploid cells. That is why it is known as reductional division. Another one is known as mitosis. This is also known as equational division. Here, the mother cell will divide to give rise to 2 daughter cells, each having the same number of chromosomes as they were there in the mother cell. That is, a 2n cell will divide to give rise to 2 2n cells. Some organisms, they may reproduce asexually by the process of mitosis. Mitosis is a characteristic feature of the somatic cells, whereas meiosis is the characteristic feature of germ cells. Now, as far as the NET is concerned, we know that as far as cell division is concerned, we are going through the entire process of cell division right from the class 8th. But what are the important things which would be asked in the NET exam or in any such competitive exams? First thing, we have to read the question carefully and look for the value of n. They would ask about a particular type of cell and what would be the result of the division, mitosis or meiosis. We also have to understand the concept of C value, because sometimes these simple concepts would be made some complicated by the introduction of term C value. For example, what is C value? C value is the total content of the DNA which is present in a diploid cell. So instead of 2n, it would say 2c, and here the main confusion will start. Now, the one more thing that normally is being asked, that we have to remember that DNA is duplicated or it is replicated in S phase. So, after the completion of the S phase, the amount of DNA would get doubled. This particular fact is being asked so many times in so many forms. Here, the trap would be the number of chromosomes, number of chromosome pairs, the type of cell, the type of division, all these things would be changed, and that is how the entire question that is being asked can take varied forms. Normally it is being advised whenever we are attending such questions, we should have a pencil and paper and we should carefully look for the value of n or c, whatever it may be, and then do it on paper and then only attempt the answers, because most of the answers would be they will appear as if the answer is correct. A cell with 16 pair of chromosomes undergoes meiosis. How many chromosomes does each of the resulting cell have? Here you can see the question is asking a cell is having 16 pair of chromosomes, undergoes meiosis. Now when it is asking for chromosome number. So we know that after meiosis a diploid cell will give rise to 4 haploid cells. So here the n is 16. So the answer would be C, that is 16. Now another question, a cell from rapidly dividing population of cells is having 2C amount of DNA at the end of mitosis. What will be the amount of DNA at G2 phase? Now we know that a diploid cell is having 2C amount of DNA. When S phase is completed and during the S phase the replication of DNA or duplication of chromosome takes place, so 2C will become 4C. So by the end of the G2 phase it will become 4C or double of the amount which is there in the mother cell. So the right option would be B, that is 4C. Here you can see again it is changing the type of the cell. If the pollen grain of a plant is having 1.8 picogram of DNA, what would be the amount of the DNA in the leaf cell? We know that pollen grains are haploid and that is would be having only N or 1C amount of DNA. Whereas the leaf cell is a diploid cell, it would be having 2N or 2C amount of DNA. So in that case, the right answer would be 3.6 picogram, exactly double as it is there in the pollen grain of a plant. So it would be 3.6 picogram, or B would be the right option. Now this question is very simple, but it is having a trap. Synthesis of DNA polymerase occurs at. We know that synthesis of DNA or replication of DNA takes place in S phase. So obviously the preparations for the replication of DNA would start in G1 phase. So the right answer would be the G1. As far as cell cycle is concerned, it is the sequence of events by which a cell duplicates its genetic material and divides into 2 daughter cells. Or in other words, we can say that the orderly sequence of events by which cells divides into 2 daughter cells is known as cell cycle. The cell cycle is having 2 main phases. One is interphase and another is M phase. Now, as far as interphase is concerned, the interphase is the preparatory phase of the cell cycle, and during this phase the cell will prepare for the cell division. So whatever is required, the replication of the DNA, the duplication of the organelles, various enzymes which are required for a cell before dividing would be generated, would be synthesized into the interphase. Whereas within the mitotic phase, the karyokinesis would take place, that is the division of the nucleus will take place, and that would be followed by cytokinesis. So most of the time cell will spend its time in interphase. If we take an example of 24 hours, what we will see, out of, suppose if a cell divides in 24 hours, for 23 hours it would remain in interphase. That is approximately 95% of the cell cycle would be spent in interphase, and the remaining 4 to 5% would be there in the mitotic phase. So in a sense we can say that interphase is the cause, it is the preparation, and the mitotic phase is the effect during which the actual cell division takes place. As far as interphase is concerned, we have already discussed that this is the phase which represents the time during which the cell is preparing for the division by undergoing various cellular growth and DNA replication through a very much ordered sequence of events. Interphase is further divided into three subphases. The first one is G1 or gap 1 phase. This is the period between the end of the mitosis and the start of the replication. And during this phase, most of the enzymes which are required for the replication of DNA are synthesized in this stage. Now the second phase, it is very crucial. It is known as S phase or synthesis phase because the replication of DNA takes place during this stage. And by the completion of this stage, the genetic material would get doubled. This particular fact is being asked so many times in so many forms. There comes G2 or gap 2 phase. This is the phase that marks period after the replication of the DNA and the start of the mitotic phase. During this particular phase, the rest of the organelles and proteins are being synthesized during this particular phase. Now, if we see in the nutshell, G1 phase also represent the time during which the cell will duplicate most of its organelles. S phase will represent the stage during which chromosome would get doubled and they would now consist of a pair of sister chromatids. And G2 phase, rest of the chemicals, whatever components, proteins are required, they would be synthesized and stockpiled. Now, these gap phases would separate the mitotic phase from the S phase. As far as S phase is concerned, the daughter cells would receive the same complement of the chromosome as the parent, and each strand of the chromosome would be used as a template to produce a new double helix of the DNA. In this particular diagram, we can see G1, S, G2 and M phase are visible in this particular diagram. Here we can also see the G0 phase of the cell cycle. We'll be discussing about the G0 phase also. As far as the cell cycle is concerned, it will start with the movement of the cell from G0 to G1, followed by S, then G2, and then in mitotic phase, the two daughter cells would be formed from a single mother cell. Now we can see certain questions. If radioactive thymidine is used during S phase of interphase, it will make chromosome radioactive near the centromere, at both the ends of the chromosome, in euchromatin region, or both in euchromatin and heterochromatin region. Since we already know that DNA replication is semi-conservative, so one strand of the DNA would be the template, and the entire new strand that would be synthesized, that is known as daughter strand, would be new. So whether it is euchromatin region or it is heterochromatin region, both would receive the radioactive thymidine via the nucleotides. So the option D would be the correct. Now, M phase. The period of actual division would be called as mitotic phase, and it will correspond to the various stages that we normally see during the mitosis. It will comprise of karyokinesis, that is the process during which the duplicated chromosomes are separated into two nuclei. This karyokinesis would be followed by cytokinesis, that is the stage during which the entire cell will divide into two daughter cells. Now, as far as G0 phase of the cell cycle is concerned, whenever a particular cell takes a conscious decision not to divide, either transiently or permanently, then it is considered to be in the G0 phase of the cell cycle. So whenever a cell withdraw from the cell cycle, permanently or transiently, it would be called as G0 phase. The cells which are in the G0 phase would not undergo any DNA replication or any kind of cellular division. The examples of the cells in the G0 phase are nerve cells and Naïve lymphocytes. Normally we don't see nerve cells dividing, and they would be in the G0 phase of the cell cycle. As far as lymphocytes are concerned, particularly the Naïve lymphocytes, they would not divide until or unless the antigen for which they are specific enters into the system. Once there is an antigen for which these lymphocytes are specific, it would provide the necessary stimulus for this cell to enter into the cell cycle. Otherwise, it will remain in the G0 phase of the cell cycle. This is a typical cell cycle, and we can see the most of the time the cell is in the interphase. G1, S and G2, it is the G1 phase during which most of the organelles would duplicate. Then in the S phase, the replication of the DNA would take place, and in the G2 phase, most of the proteins would be synthesized in the G2 phase. That would be followed by the M phase or the mitosis phase. The mitosis phase is further divided into prophase, metaphase, anaphase and telophase. This would comprise the karyokinesis, and after that cytokinesis would result into the generation of two daughter cells from the single mother cell. Now, this particular diagram shows the movements of the chromosome or the chromatids during the mitosis phase. The mitosis phase are being categorized into 4 stages known as prophase, metaphase, anaphase and telophase, based on what is happening, what are the movements which are taking place within the cell nuclei. So within the prophase, the chromosome would start condensing, the nuclear membrane will disintegrate, the nucleolus would disintegrate. That would be followed by the metaphase, during which the chromosomes are most condensed, and that is why metaphase is the preferred stage for the studies of chromosomes. Any kind of bending studies or the studies of the mutations of the chromosomal segments are being done in the metaphase only because of the fact that chromosomes are most condensed at this stage only. Besides this, the chromosomes are arranged at the equatorial plate during the metaphase, and this is the most characteristic feature of the metaphase. By the start of anaphase, these chromosomes which are arranged at the equatorial plate would start moving towards two opposite poles, and by the end of the telophase they would reach the 2 poles, and that would be followed by the cytokinesis. In this diagram we can see the happenings which takes place in the S phase, prophase, metaphase. We can see the arrangement of chromosome at the equatorial in the metaphase. That is followed by anaphase, where the movement of the chromosome towards opposite poles starts, and by the telophase they reaches the opposite poles and the nuclear envelope would start resynthesizing. Most of the events that takes place in the telophase would be exactly opposite of the prophase, and this would be followed by the cytokinesis, resulting into the daughter cells. Now, one of the important fact is how cytokinesis takes place. It differs in animals as well as plant cells. The cytokinesis is the process by which the entire cell is being generated from the single mother cell. In the case of animal cells, cell furrows are being formed from the periphery, and that will move towards the center. So, it is by cell furrowing or invagination that animal cell will divide. Whereas in the case of plant cells, it would be by cell plate method, where some certain substances would get accumulated at the periphery, and this accumulation will increase and will spread towards the periphery from the center, and this process is known as cell plate method. So plant cells and animal cells, they would be having different kind of cytokinesis. In the animal cells, it would be from the periphery towards the center, whereas in the plants it would be from the center towards the periphery. So this particular fact is also being asked in the exams. So the questions that are normally being asked are, by which method plant cell divides, that is cell plate method. By which method animal cell divides, by invagination or furrows. Which cell divides from periphery to the center, the animal cell. Which cell will divide from center to the periphery, that is the plant cell. So these particular facts may be asked without naming that it is a plant cell or animal cell. For example, one may ask a particular leaf cell is dividing. It will divide from center to periphery or periphery to center. So we have to keep in mind that leaf cell is a characteristic plant cell. So obviously it will be from center to the periphery. Now we also have to keep in mind that from the division point of view, cells are categorized into three. Cells which normally do not divide, for example, nerve cells, muscle cells, or RBCs. Now, they would not give any hint, they will just ask, nerve cells are normally found in which stage of the cell cycle? Since we know that nerve cells, muscle cells, and RBCs, they do not divide, so they normally remain in the G0 phase of the cell cycle. This fact has been asked so many times. There comes the cells that do divide occasionally. There would be a stimulus for them. For example, when the antigen is present, the lymphocytes will divide. Likewise, whenever liver regeneration is required, the liver cells will divide. So there are cells which divides occasionally and they will enter into the cell cycle whenever there is a right stimulus or whenever there is a need. For example, if there is some wound, the nearby cells will divide to fill that particular wound. There comes actively dividing cells. These cells are normally constitutive, their activities are required too much in the body, and they will be high on as far as mitotic activity is concerned. The example of such kind of cells is spermatogonia that give rise to male gametes or the sperms, the hematopoietic stem cells that give rise to the various kinds of blood cells in our blood, and the cells of the apical meristem. Sometimes these facts are also being asked, which particular cells are normally active in the cell cycle. There are certain questions which are also being asked on certain alkaloids. There are certain alkaloids like colchicines which have been exploited like anything in various cell studies and even in the increasing the ploidy level. So here we see a particular question. The colchicine treated cells are arrested in S phase, prophase, G1 phase or metaphase. Actually, colchicine will interfere with the mitotic spindle fiber formation, and even though the chromosome has replicated, they have doubled in their number, but they will not divide into, they will not get distributed into the daughter cells. So the ploidy would get doubled without cell division taking place. So it is the metaphase during which the colchicine will show its effect, and that is why would bring about the doubling of the chromosome. Since colchicine has been involved in the generation of Raphanobrassica and various studies of the cell cycle, such concepts are normally being asked in the exams. Now we can see again the same kind of jargons are being used to confuse the students. The DNA content in a pollen mother cell is 2C. What would be the content of DNA per cell at pachytene? Now we know that pollen mother cell would be dividing by meiosis, and the meiosis will give rise to, at the end of the meiosis 2, it will give rise to 4 haploid cells. But before that, by the start of the meiosis 1, cell would already be having double the amount of chromosome or the genetic material. So 2C by the end of S phase will become 4C. In the pachytene, crossing over takes place, but chromosomes are not separated. So the amount of DNA would remain double as it is there after the completion of the S phase. So the right answer in this particular case would be the C, that is 4C. And by the end of the meiosis 2, it will become 1C in each of the haploid cells. So just by changing the stage, for example, if it asks what would be the content of the DNA per cell after the completion of meiosis 2, it would be 1C. But if it asks any stage, suppose it is pachytene or diplotin, it will remain 4C. Any stage after S phase, the answer would remain 4C. Another question that we can see here, the major events of mitotic prophase includes all of the following except chromosome coiling, DNA replication, nuclear envelope breakdown, or nuclear disaggregation. We know that the DNA replication takes place in the interphase, particularly the S phase. So the B option is correct. This is the event of the replication that takes place in the interphase, and it does not take place in the prophase. Rest of all these things takes place in the prophase. Now, the basis for the blocking action of the alkaloid colchicine on the cell division is the stage would be M phase, and the basis would be that it interferes with the spindle fiber formation. Now, in a dividing cell, DNA synthesis goes through only one round and is arrested before cytokinesis. The arrest is due to. Now, this is the first question that is giving us the first-hand experience of the cell cycle regulation. Specific cyclin proteins along with CDC2 protein kinase, CDC2 protein kinase, G proteins with CDC2 protein kinase. The correct answer would be B. As we will see in our discussion, that specific proteins whose concentrations rises and falls in a very cyclic fashion are known as cyclins, and these cyclin, via their impact on specific protein kinases, and since these protein kinases are dependent on cyclin, they are also known as cyclin dependent protein kinases. So cyclins along with cyclin dependent protein kinases, of which CDC2 is an example, present in yeast and some lower eukaryotes. So the correct answer would be B. Now, this is a very simple question. How many DNA molecules are present in the nucleus of human somatic cell in G2 stage of the cell cycle? We know that there are 23, the N in the human is 23, we are having 46 chromosomes. So after the completion of the S phase, the 46 would become 92. So in the G2 stage of the cell cycle, the number of DNA molecules would become 92. So D is the right answer. Another question, during embryonic development, the total cell cycle time for a given cell type can change. These changes are primarily due to variations in G1 phase, G2 phase, M phase or S phase. Here it is remarkable that different species are having different cell cycle time. It is also called as generation time. That is the time that is required by a cell to complete its cell cycle. So when a cell starts its division to the time when it completes the division, this particular time period is known as cell cycle time or generation time. For different species it is particular. So mostly we see that it is the G1 phase, that is during which the most of the preparations of the cell cycle are going on. It is different in different cases, and most of the variations would be confined to G1 phase. Now we are going to discuss about cell cycle regulation. As far as our discussion is concerned, till this particular time, we have seen the transition of the cell from one stage to the next, that is interphase and mitosis phase. Within the interphase, G1, S, G2, within the M phase, prophase, metaphase, anaphase, telophase, followed by the cytokinesis. Now, whatever cell type you take, these sequence of events remains the same. So it was a question obvious that what is regulating and why it is not like that ki G1 comes after G2, S comes before G1. What is regulating all these stages of the cell cycle? So people were interested in looking into what are the molecules, what are the enzymes or proteins which are involved in the regulation of the cell cycle, which are managing orderly sequence of the events which are the characteristic of any cell cycle. So these cell cycle events are obviously triggered by one or the other cell cycle control system. There were certain sets of experiments by which it was clue was being obtained that there might be certain set of molecules which are found in the cytoplasm, which might be involved in the cell cycle regulation, and these set of molecules and their activities might be affected by internal or external controls or the signals. This is very interesting because those people or those scientists who are working on the cell cycle regulation, they didn't knew that their outcomes or their finding is going to play a very important role in the treatment of the cancer. Because during that particular era, cancer was being studied differently, and people who were interested in the pure sciences or in the studies of the cell cycle stages, that was different. But our findings into the cell cycle regulation brings new insights into the cancer treatment. How it is being governed, that we'll see as the discussion will unfold. So there are certain molecules which triggers the cells to enter a stage in the cell cycle. These particular molecules which are involved in the transition of the cell cycle from one stage to the next are known as maturation promotion factors and cyclins. There are certain questions which are being asked on the cell cycle. The cell cycle is controlled by proteolysis of cyclin-dependent kinases, phosphorylation of the cyclins, dephosphorylation of the cyclins, and the proteolysis of cyclins. Here it is worth mentioning that the levels of the cyclin-dependent kinases are constitutive or they remain same. They are normally not being proteolized. The limiting factor is the concentration of the cyclin. Cyclin is synthesized, reaches, as there is a buildup of cyclin, the activity of cyclin-dependent kinases increases, and by the time a cell reaches the M phase, the concentration of the cyclin is maximum. Thereafter, cyclins are degraded, their concentration falls, and the fall in the concentration of the cyclin will result into the fall in the activity of the cyclin-dependent kinases. All these things now we are going to discuss in the part 2, that how cyclins would have their impact on cyclin-dependent kinases, how many types of cyclins are there, how many kinds of cyclin-dependent kinases are there, what is the interdependence of the cyclins and cyclin-dependent kinases, and are there some more molecules which might be considered superregulators which regulates the entire cell cycle. This we'll be discussing in the part 2. Thank you.
Vande Gujarat.